Hello and welcome to Sciencera. In this video, we're going to discuss DNA transcription. DNA transcription, also known as RNA synthesis, is the process by which genetic information that is contained in DNA is rewritten into mRNA by an RNA polymerase enzyme. The synthesized mRNA is transported out of the cell nucleus where it will later aid in synthesis of protein by mechanism of translation. Regulation of mRNA production in nucleus, the cell automatically regulate the rate of gene expression. Process of transcription is aided by the RNA polymerase enzyme which copies the right sequence on DNA template to produce a complementary RNA copy of gene. The basic mechanism of transcription is same in both eukaryotes and prokaryotes. However, it may differ in several ways between them. DNA transcription enzymes and function. The major enzyme used in DNA transcription is RNA polymerase. In prokaryotes, one type of RNA polymerase enzyme is used, while in eukaryote, three type of RNA polymerase enzymes is used. For example, RNA polymerase 1, 2, and 3. The major function of RNA polymerase enzyme includes formation of initiator complex which aids in unwinding process of the double helical structure of DNA, synthesis and elongation of RNA transcript by adding the nucleotide bases like adenine, cytosine, guanine and uracil. It forms the termination sequence that stops and terminate transcription. Let's look at DNA transcription, also known as RNA synthesis. Steps of transcription include 50 different protein transcription factor will bind to the promoter sites on the 5' side of gene to be transcribed. The RNA polymerase binds to the transcription factor complex allowing the DNA helix of DNA to open up. RNA polymerase then reads one strand in 3 to 5 direction. In eukaryotic cell, the nucleosome in advancing RNA polymerase for protein coding genes. The RNA polymerase and transcription factor complex replaces the nucleosome after DNA has been transcribed and POL2 has moved on. As the RNA polymerase moves along the DNA strand, it assembles ribonucleotides into a strand of RNA by utilizing triphosphate or ATP. Each ribonucleotide is inserted into the growing RNA strand by a base pairing. For example, each cytosine is linked to guanine while a uracil is linked to an adenine. The RNA synthesis takes place in 5 to 3 direction. As each nucleoside triphosphate is added to 3 end of the growing strand, the 2 terminal phosphates are removed. When transcription comes to an end, the transcript is released from the polymerase and polymerase enzyme is also released from DNA. Let's look at the DNA transcription process. In prokaryotic cell, the entire mechanism of transcription is summarized in 3 stages: initiation, elongation, and termination. At the end of termination in prokaryotes, the mRNA formed is ready for translation. Unlike in eukaryotes, after termination, an immature mRNA is formed and therefore more processes are needed to form a mature mRNA which is translated into proteins. Generally, the transcription process transcribe DNA into mRNA, the type of RNA that carries the information that is needed in the synthesis of proteins. In eukaryotes, there are two broad steps that take place in transcription. Number one is pre-messenger RNA formation using an RNA polymerase enzyme, and number two is editing of pre-messenger RNA by splicing. Pre-messenger RNA formation involves the initiation, elongation, and termination phase, which ends by forming the mRNA. mRNA then undergoes different stages of splicing to form mature mRNA. Let's see how formation of pre-messenger RNA occurs. When transcription starts, DNA must unwind aided by RNA polymerase enzyme which catalyzes the process. In transcription, only one of the DNA strand is transcribed, the strand that has initiator sequence. This strand is known as the sense strand, while the complementary strand is known as the antisense strand. mRNA that is transcribed is normally a copy of sense strand, however, it is antisense strand that is transcribed. The ribonucleoside triphosphate or NTPs align along the antisense DNA strand by base pairing. Then the RNA polymerase joins the ribonucleotide together forming a pre-messenger RNA molecule complementary to a region on the antisense strand. Transcription is completed when a RNA polymerase enzyme finds a triplet of bases read as a stop signal. At this stage, the DNA molecules rewinds to reform the double helix. The formation of messenger RNA is done in three stages: initiation, elongation, and termination. Promoter and initiation in prokaryotes. The initiation and transcription is signaled at a region known as a promoter. The promoter is the site of RNA polymerase binding such that the promoter guides the polymerase where it should sit on the DNA in order to initiate transcription. RNA polymerase is the enzyme that catalyzes the mechanism of transcription. RNA polymerase enzyme has a sigma factor, which is this associative unit that allows the enzyme to recognize the promoter sequence. Starting point of transcription, which is spaced between -35 and -10 region, as you can see in the diagram. The promoter sequence, promoter and initiation in prokaryotes. The promoter sequence is recognized by RNA polymerase holoenzyme subunit by attaching to and moving along the DNA template molecule. This forms a closed promoter complex. Single DNA molecule may have multiple promoter sequence or closed promoter complexes. The promoter, which is bound with transcription factor along with RNA polymerase, forms a complex. Transcription factors are regulatory proteins that control transcription rate. When the RNA polymerase is bound to promoter sequence, it denaturalizes the DNA duplex locally, forming open promoter complex which become unwound part of the double-stranded DNA, exposing the base on each of the two DNA strands. Promoter and initiation in eukaryotes. In eukaryotes, the RNA polymerase does not directly attach to the promoter sequence like prokaryote. A helper promoter known as basal or general transcription factor binds to the promoter first, which helps the RNA polymerase attach to the DNA template. Eukaryotes have a promoter sequence called Tata box, which is recognized by transcription factor, which eventually allow the binding of RNA polymerase. The Tata box has lots of A's and T's, making it easy to pull the strand of DNA apart. Elongation. After initiating transcription, the sigma factor dissociates from the RNA polymerase. The template strand is read in 3 to 5 direction, which means that RNA synthesis take place in the 5 end to 3 end direction, with nucleoside triphosphate acting as the substrate for the enzyme. The other strands from the DNA template is known as the coding strand. Because the base sequence of the new mRNA is identical to it except for the replacement of thiamine with uracil base. The RNA polymerase catalyzes the formation of phosphodiester bond between the adjacent ribonucleotides. Energy used by RNA polymerase is derived from splitting the high-energy triphosphate to monophosphate, releasing the inorganic diphosphate. A transcription bubble is formed and it must be maintained since transcription takes place on the double-stranded DNA template. The bubble moves along the DNA duplex during the elongation. Stalling or pausing are common, which are later essential for transcription termination. Let's look at termination. This is the process of ending transcription which happens when signaled by a stop sequence known as a terminator sequence. This happens when the RNA polymerase transcribes the terminator sequence. RNA polymerase then releases the DNA template which unwind back to a double helical structure. Termination in bacteria. Termination are of two types: RHO dependent termination. This is the termination process where the RNA molecule contains a binding site for a protein known as a RHO factor which binds to a DNA sequence. It starts to climb up the transcript towards the RNA polymerase and reaches the transcription bubble. At the bubble, the RHO factor pulls the RNA transcript and the DNA template strand apart, releasing the RNA molecule and terminating the transcription process. A transcription stop point sequence that is found later in the DNA causes the RNA polymerase to stop and allow the RH factor to catch up and terminate the process. The second type of termination in bacteria is RHO independent termination. This process depends on a specific sequence found on the DNA template strand during the transcription. As the RNA polymerase approaches the end point of generally that is transcribed, it reaches a region that is rich in cytosine and guanine. RNA that is transcribed from this region folds back on itself and complementary C and G binds together, forming a stable hairpin that makes the RNA polymerase to stall. The hairpin is followed by uracil in the RNA terminator, which is complementary to DNA template adenine. The UA region forms a weak interaction with DNA template and with stalled RNA polymerase cause an instability, allowing the enzyme to fall off and end from the new RNA transcript. Pre-translational mRNA processing. In eukaryotes, the mRNA that has been transcribed is known as pre-mRNA and therefore it must undergo other process for it to mature into the mature mRNA. These are known as pre-translational mRNA process. They include number 1, 5' capping. This is the addition of methylated guanine cap to the 5' end of mRNA. The 5' cap helps in the recognition of mRNA molecules by ribosomes and to also protect the immature mRNA from degradation of RNA. Next is polyadenylation. This is the addition of poly-A tail to the 3' end of mRNA. The poly-A tail is made up of several molecules of adenosine monophosphate which stabilizes RNA because of its natural instability. Last is splicing. This is the coding of one genetic sequence for different protein, conserving the genetic material. The process involves removing of known coding sequence known as introns by spliceosomes excision, joining of coding sequence known as exon by ligation. Splicing is sequence dependent, therefore it occurs within the transcript. This allows many protein to be made from a single pre-mRNA. At the end of the splicing process, mature mRNA will have been made. Mature mRNA then becomes the messenger carrier which allows protein synthesis to occur. Mature mRNA has open reading frame (ORF), a region that gets translated into proteins. Translation on the ORFs is done in 3 blocks of 3 nucleotides known as codons. At the end of 5' end and 3' ends are untranslated region UTRs which are not translated during the protein synthesis. Reverse transcription. This is the conversion of RNA to DNA by which RNA act as the template in synthesis of a type of DNA known as the complementary DNA. The central dogma defines the mechanism that involves DNA synthesis, replication, RNA synthesis, transcription, and protein synthesis, translation, and cDNA synthesis, reverse transcription. Therefore, DNA codes for RNA, RNA codes for protein, and RNA can also code for DNA in case of reverse transcription. RNA encodes viruses undergoes that reverse transcription mechanism where their genomic RNA is converted to DNA by aid of the reverse transcriptase enzyme. Reverse transcription is also known as a retrotranscription or retrorase. It is an extremely erroneous process that can lead to mutation that can cause drug resistance. The conversion of RNA to DNA is normally desirably applied in laboratory majorly as a diagnostic tool for most RNA viruses such as HIV or hepatitis. Transcription inhibitor. Transcription inhibitors are the elements that are used to inhibit the action and mechanism of RNA polymerase enzyme, which hinders the process of transcription. Transcription inhibitors are used majorly to hinder bacterial mechanism of transcription in disease-causing pathogen. Some of the most used inhibitors include alpha-amanitin. This is an inhibitor extracted from yeast that is selective for RNA polymerase II and RNA polymerase III. Next is rifampicin inhibits bacterial transcription by inhibiting DNA-dependent RNA polymerase by binding to the beta subunit. Number 3 is 8-hydroxyquinone is also an antifungal transcription inhibitor. Other includes actinomycin D, CDK9 inhibitors such as DRB and flavoperidol or triptolide. An inhibitory mechanism is histone methylation that bars the action of transcription. This brings us to the end of RNA synthesis. Thank you for watching. If you like my video, don't forget to subscribe for more.